I'm going to talk about collecting evidence in Parkinson's, but really particularly with a focus on genetics and what genetics can tell us about the variety of Parkinson's patients and what sort of treatments they should be taking, and hopefully how this will help us develop curative treatments in the future. I think there's been a huge amount of development in the work that we've been doing on a collaborative basis with researchers across the world over the last 10-15 years. I think we made some great advances in understanding the biology of Parkinson's. The problem is that so far that hasn't translated into new treatments for patients. So there's a gap between what we've learned in the lab and in our collaborative analysis and actually converting that to new patients. So I think, obviously coming to speak with this group, my clinical practice, we're aware that there is a huge pressure now to use all this information and actually all this money that's been spent on genetic research and to harness benefit for patients. And I think I am optimistic that we will see a lot of progress in this area over the next 5-10 years. I think there are two sort of aspects to this actually. One is that our ability to analyse people's individual genetic makeup, to correlate that with their clinical symptoms, to understand the biology of why they've developed Parkinson's is progressing extremely quickly. But also I think we're entering a new era of treatments in which there are some very powerful biologic treatments that are coming along, which I think are going to work in different ways to some of the treatments that we've had before for Parkinson's. And obviously Cure Parkinson's Trust has done a fantastic job in stimulating the amount of clinical trial activity, the number of clinical trials that are up and running, and the ones that actually, clinical trials are actually more logically directed towards the biology of Parkinson's. So it's going to be very much focused on genetics. It is, I think, very complementary to Roger's talk, really talking about the heterogeneity, the variety of Parkinson's disease. I'm going to talk about genetic data in research. So yes, as Richard said, I'm a neurologist at Queen Square in the Royal Free. I spend about half my time doing research, primarily genetic research, increasingly clinical trial based research, and half my time doing clinical practice. So in terms of the talk, I'm going to give an overview of what I think is the sort of state of the art in Parkinson's genetics research, so where we've got to and what we understand about things currently in terms of Parkinson's. I'm going to talk about how we can translate that into new treatments and give some examples of what's happening in terms of using this work to develop new treatments for Parkinson's patients. I'm going to talk a bit about ongoing genetic studies. I'm going to talk about something which I think is extremely important and one of the reasons why I'm kind of keen to do these types of talks with patients and people affected by Parkinson's, which is the issue of communicating results, understanding results, and how we communicate what we're discovering, both on a sort of global level, but also on an individual level in terms of understanding the basis of your condition. And then I'm going to finish off with a sort of manifesto or plan of action for some of the things I'd like to see happen in terms of developing this area over the next 5 to 10 years, which I'm sure we will all be taking forward, both as researchers, doctors, patients, Cure Parkinson's Trust, people who are funding research. I think we'll be working together to take forward this plan of action. Okay, so in terms of the state of the art, the key points, and actually hopefully I'm not going to go into infinite detail on all these different markers and genes. I'm going to give a few examples, but it's more going to be about the principles of things really, rather than about nitty-gritty. This is designed as a lay talk. I'm aware that there's some people here who are extremely knowledgeable about some of these areas who may want to ask me some more detailed questions. So I'm obviously happy to discuss, but this is really designed as a kind of overview talk to talk about the principles of things, rather than to go into huge depth on research data. So I think key points are, first of all, key point number one is that although I spend almost all my time, or a lot of my time, doing genetics and genetic-orientated research, Parkinson's in general terms is not an inherited condition. It's usually not something that runs in families. For the vast majority of people with Parkinson's disease, there's not a hugely increased risk to other family members, and I think that's a very important thing to state from the outset. Secondly, in terms of what we have understood about Parkinson's, we have understood that there are some familial forms of Parkinson's. So occasionally, how do I know if I've got a familial form of Parkinson's? Usually that's because other people in your family have been affected. So usually when we see people in clinic, they do not have relatives who've been affected by Parkinson's. Occasionally we come across families where 5, 6, 7 people in the family have all been affected by Parkinson's. And clearly in that situation, it's very likely there's a genetic factor which is contributing to the development of Parkinson's. So that's familial genes, genes which cause Parkinson's pretty much without environmental factors other than age. So you have to live to a certain age to get the condition, but they are what I would call causative for Parkinson's. We then have what I would call sort of strong risk factor genes, so genetic variants that significantly increase your risk for disease and greatly increase the risk, but not sufficient in on their own to cause Parkinson's. We then have sort of weak risk factor genes, so things that vast numbers of the UK population have. Most people in the UK population don't have Parkinson's, but we know from doing very large studies that they slightly increase your risk. And some people say actually it's not really worth looking at these genetic variants that slightly increase your risk because they don't have a very big impact on risk. But actually we think it is important because it allows us to understand the biochemistry, the chemical pathways that are leading to the condition, and hopefully that will be a route into new treatments. There's then what I would call phenotype genes. So we know those. So these are genes that or genetic variants that change the way that you might respond to treatment or to change the way that to change your disease course. So we know in Parkinson's that some people the disease is very stable, doesn't change that much over 10, 20 years. People develop more problems as time goes on, but at quite a slow rate. Other people develop more problems, in the first year or two that they have the condition. And we haven't really focused on that this much up to this point, but actually there is a huge variation in the progression of Parkinson's. And one of the things we're working on, Manuel is here, is one of our PhD students who's working specifically on this, is actually try to understand what the basis of that is. So obviously if we knew what the genetic variant was that makes one person have a very benign form of the condition and another person a much more severe form, if we knew what that genetic variant was, and we could make a drug to replicate that effect, we would have an excellent treatment for slowing down the change over time. So understanding that variation between patients we think is important. And then lastly, I think one of the things which is now starting to open up, and again we're starting to do some research on, is actually going beyond genes and looking at people's chemical makeup in blood samples or even in spinal fluid samples to understand what's happening with their condition based on the chemical makeup in their blood. And actually, before I go on, perhaps I'll just say a bit about genetics as a sort of reminder for people. So, in terms of what a gene is, we all have about 20,000 genes. We have 2 copies of each gene, one from our mum, one from our dad, and the genes contain the instructions that really control all of the cellular processes in all the cellular processes in the body. Obviously, when we have children, when we make sperm or eggs, we only hand on one of each of our genes. So we get one from our mum, one from our dad, and if we have children, we only hand on one of our 2 genes. So that's the basis of passing on genetic information and inheritance. We know that if we went right now and took a blood sample from everyone here, we could take your blood sample to the lab. We could chomp up the white cells and extract the DNA from your blood sample, and we could sequence your genetic makeup. So we could look across all of the letters in the genetic code, all of the 20,000 genes that make up your genetic makeup. We know just about every 1,000 letters through the genetic code there'd be a variation. So we'd see a difference in people in the room based on looking at their genetic makeup. So a lot of this variation in the genetic code has no effect at all. Some of it accounts for normal variation between people, so height, hair colour, sporting ability. Many of these things we know run in families, and we know that that's genetically encoded. Some of these variants are important in causing risk for disease. So these weak risk factor genes, as I've referred to. So these don't in themselves cause a condition, but they increase your chance of getting it. So an example of that would be a cholesterol gene that slightly raises your cholesterol levels. So that doesn't cause you to have a stroke or a heart attack. But if you carry that genetic variant and you eat a very high cholesterol diet, then the two things together will cause, and you have other risk factors like high blood pressure, then those things together will lead to you having a heart attack or stroke. So those are risk factor genes that many people in the population have, but most people don't develop the condition of interest. And then these are very rare variants in the genome that actually cause the condition. So if you get that variant and you live long enough, you will get the disease. So those will be strong risk factor genes. So that's kind of the basis of genetic variation. And our ability actually to sequence the genome and to understand rapidly variation between people has changed very quickly. So, in the space of an afternoon, we can look at 500,000 variants across the genome and compare them between people with and without Parkinson's or any other condition that we want to look at. So actually the technology for this has changed very quickly. Our ability to generate data has changed very quickly. What I think is probably fair to say is changing more slowly and is more of a challenge now is actually handling the data, having powerful enough computer systems, having enough storage, having the statistical know-how to actually analyse such vast amounts of data that we're now generating from patients with Parkinson's. And what we're particularly interested in doing now, of course, is not just looking at whether someone has Parkinson's as compared to someone without Parkinson's, but actually looking at the type of Parkinson's someone has. So look at their drug response, whether they get dyskinesias, so levodopa-induced dyskinesias, look at whether they develop balance problems, whether they have bladder or bowel problems, which some Parkinson's patients get, but not all patients, to understand this variation between people and actually link up the clinical information with the genetic information. And again, in order to do that, what we really need to do, and actually when we started doing these studies, so the first UK study looking at Parkinson's risk across the whole genome was published in about 2007, and that involved 1,700 Parkinson's patients and about 6,000 people without Parkinson's. We thought we looked at that. We made some discoveries of things that were different between Parkinson's patients and people without Parkinson's. And after that, we thought, great, we'll look at variation now between people, progression and balance problems, and look at the things that control that. But actually, the clinical information we had was very patchy and had been collected in a very inconsistent way. So that led us on to set up some new studies, which then would very systematically collect information on people. So I think that probably some people in the room have very kindly, volunteered to be involved in some of our studies, which is great. And obviously, we really appreciate everyone who gets involved in our studies. But this idea of systematic collected paper, collecting data, means that when you join the study, you'll likely be given a pamphlet with about 50 pages of questions to fill in, to try to collect as much information as possible about the condition, how it's affected you, your personal experience, and to be able to factor that in down the line into understanding what the basis is for this variation between people. So we're trying now to very systematically collect information. And that does mean, as I say to everyone who gets involved in our research, we're going to end up asking you quite a lot of questions about things that may never affect you. So we're going to ask you about everything we can think of, but that, just because we're asking questions about this doesn't mean that this is something that's going to affect you with your condition. So we're trying to, get a very broad and systematic picture of how the condition affects an individual patient. And the good thing about this, of course, is that around the world, people are using similar questionnaires or identical questionnaires and instruments, and it means down the line we can put all the data together and do very large studies to try and tackle some of these questions in the future. So in terms of state of the art, in terms of familial genes, we've now identified about 7 or 8 genes in which genetic variants inherited in these genes actually will cause Parkinson's. So the commonest genetic variant is the LRRK2 variant, the G2019S variant, which affects about 5% of people with familial Parkinson's. So you only need one copy of the gene to develop the condition. So that means if you have the gene variant and you have Parkinson's, then each of your children individually will have a 1 in 2 chance of inheriting that genetic variant. But of course, just because you have the genetic variant, it doesn't mean you will develop the condition. We know that some people live until their 70s, 80s, 90s with this genetic variant and never develop any problems. But it clearly greatly increases your risk. And if you do carry the genetic variant by about age 75, 80, a lot of people with this will have developed Parkinson's. So we know this is a, in the 3 billion letters in the genetic code, this is one letter that's altered, and this is sufficient to cause Parkinson's plus age. So this is sufficient to cause Parkinson's plus age. And that kind of, gives you, that's why, in a way, I started out doing genetics, because this is such a powerful way to understand the condition. So when I see people in clinic, when someone's affected by this, the first thing that people are, think is, why has this affected me? Why is this condition affecting me? Was it the road, was it whiplash injury I had? Was it because we, lived next to a chemical factory? Was it because we lived, in a rural existence? And actually, all these environmental factors, it's extremely difficult to do research on these, because you have to look back across someone's whole life and compare it with someone who doesn't have Parkinson's, and look at the environmental factors that may cause the disease. And actually that type of research, although undoubtedly it's important, is extremely difficult to do. For genetics, fortunately, you carry through life the same genetic variants that you were born with, pretty much. So actually we know what someone's genetic makeup was 20 years before they developed clinical Parkinson's, when the disease was likely starting to develop. And this gene variant is sufficient to cause the disease. And so that's incredibly powerful, understanding why the disease happens in those patients. And as soon as we find a gene variant like this, what we do is put into a cell model or an animal model and use that as a test bed for treatments. So both university and industry researchers running hundreds of thousands of different compounds, looking for ones that will interfere with the change in biochemistry that is related to this gene variant. In fact, this LRRK2 gene encodes a kinase, so it adds phosphate groups to other molecules. And actually, we think that this variant that causes Parkinson's increases the rate of phosphorylation, increases the rate of phosphate being added to things. And actually, for 30 years, drug companies have been making kinase inhibitors. So that is drugs you can take by mouth that switch off the kinase activity and prevent that thing from happening. So this has taken quite a long time to work all this out, but very simple research where you can see a gene variant, it makes a gene slightly overactive. Hypothetically, you have a drug that tackles that gene variant, that kinase activity, turns it off, and perhaps stops the disease in its tracks. So actually, I think there are at least 5 companies that are developing these sorts of drugs at the moment. One of these companies is Denali, and they announced their results that showing that they had what they call target engagement. So the drug did what they expected to do in terms of the kinase activity a few months ago, and that's progressing forward into clinical trials. So this is, although this is a rare situation, obviously I think it's incredibly powerful in understanding disease biology and helping us develop new treatments. So the next sort of level of genetic variants, if you like, is what I would call strong risk factor genes. So the one that we know best is the glucose cerebrosidase, the GBA gene. This is a gene variant where if you get 2 copies of this, you have a childhood disease called Gaucher's disease, which is quite rare. But one of the discoveries that's made maybe about 10 years ago was that the parents of children with Gaucher's disease had an increased rate of Parkinson's. And it turns out if you carry 1 copy of this GBA variant, you have an increased risk of Parkinson's. But this is a risk factor gene. So actually, in the UK population, about 1% of people will carry this variant but do not get Parkinson's. But if we took blood samples from Parkinson's patients, about 4% of Parkinson's patients carry this risk factor gene. And in fact, I think this is quite confusing for doctors and for patients actually. So this is a risk factor. Other factors need to be there to develop the condition. Most people who have this risk factor will never get Parkinson's. And in fact, we've just completed a study in the Proband Tracking Parkinson's study, 2,000 UK patients. When we looked at the Parkinson's patients who carried these GBA variants, they had no increased family history of Parkinson's. So this wasn't something that was causing massively increased risk through the family. But again, and I'll say a little bit more about this in a few slides, but again, this happens to point for one particular pathway within cells. We know that this variant, this pathway, is very strongly associated with Parkinson's. So this kind of points us towards a therapeutic target. If we can get a drug that restores this GBA function, then we'll have something that is a useful therapy for Parkinson's patients. So then moving down slightly, we're talking about weaker risk factor genes. So these are the sort of genes or genetic variants that we need. Actually, we're up to now in a sort of global consortium, we're up to about 20,000 Parkinson's patients as compared now to 100,000 plus controls. So we're up to very, very large numbers to find these genetic variants that have a very, very small effect. We've identified so far about 40 of these genes. Individually, I don't think it's very useful to know currently whether you carry one of those gene variants. I'll say more about that in a few minutes. And the reason for that is that actually in this particular gene variant that I picked out here, the tau gene, the variant that actually causes the risk for Parkinson's is carried by about 75% of the UK population. But if you look at Parkinson's patients, it's carried by about 85%. So it has a relative risk of about 1.2. So it's a tiny increase in Parkinson's risk. The vast majority of people, again, with this variant will not develop Parkinson's, but it gives us some clues into the mechanism which leads to the condition. So then, as I mentioned, phenotype genes. So this is something we're starting to explore. So we know that one of the things that affects particularly younger patients with Parkinson's is susceptibility to side effects to medication. So dyskinesia, so involuntary movements in response to medication is one thing. Another thing that is common with drugs like pramipexole, ropinirole, rotigotine is this impulse control disorder, which many of you who will have been talked about those drugs will have heard about this. This is a sort of abnormal drive which leads people to spend lots of money, gamble, download lots of stuff from the internet. So this is ICD, impulse control disorder. We think that these things are probably genetically determined. So actually people who are susceptible to this have a family history of alcohol abuse or gambling. So we think it's likely these things are genetically determined. If we can pinpoint these genetic variants that increase the risk of this before we give people medication, we'll be able to give them a better idea of whether they'll get side effects. So if we know what side effects you're going to get, or can predict to some extent what side effects you're going to get before the medication, obviously that enables us to give much better treatment to people, and on an individual level allows us to tailor treatment to people much more accurately. So I think this is the kind of in a way is one of the next frontiers that I'm particularly interested in, understand the variation between people and how that relates to genetics. So lastly, biochemistry, chemical makeup. So some of the work that Phil Campbell is doing in our group at the moment is just taking blood samples from people, taking rather large amounts of blood from people with Parkinson's, and looking for blood signatures that might indicate that they have problems with mitochondrial function. So that's the energy supply in nerve cells. So that's actually ignoring now the genes, but going straight to the chemical profile that you can measure in blood, and looking to see if there's a subset of patients with Parkinson's who have mitochondrial problems. And those patients presumably would respond well to a mitochondrial therapy. So as a kind of an aside, which really Richard referred to and Roger's going to refer to as well, but many of you may have heard of coenzyme Q10, which is a drug, effectively kind of souped up vitamin that improves mitochondrial function. There's been a very large trial in Parkinson's patients, thousands of Parkinson's patients given this medication compared with placebo, compared to the control treatment, and there was no difference in the rate of progression, the change over time in patients with mitochondrial treatment as compared to patients without the mitochondrial treatment. But it may be that only 20% of patients will have responded to it. So maybe if we picked out the right 20% of patients and given those patients the coenzyme Q10, they may have done really well with that. And so maybe the lack of ability to pick out patients who are going to respond to treatments is a kind of barrier to our future development of treatments. So we're starting to, this is something genetics is quite easy to do because your genetic makeup, actually handling DNA is quite stable. Someone can take a saliva sample in Africa, can put it in the post, send it to us, we can send it to the lab, and we can extract the DNA, look at someone's genetic makeup. So it's a kind of pretty stable thing to do. The things that we're now doing, looking at chemical makeup in blood, we have to take approaching 100 mils of blood. There has to be someone who take it to the lab that day and run all the assays that day, within an hour of the sample being taken. So that's kind of laborious, and clearly there's obviously going to be a time delay before that type of approach will be translatable in something that can be done in any clinic as an easy test to find out what sort of treatment you want to take. You're likely to be respond well to. But I think that this will change very quickly, and we've seen technology change very quickly in genetics. I think we'll see technology changing very quickly in this sort of study of proteins in the blood, proteomics, to use a sort of fancy term, and metabolomics, looking at metabolic profiles in blood. I think this will change quite quickly, and I think that it will become much more useful as a way to predict what sort of treatments people are going to respond to. So translating this into new treatments. So we've done all this work. Patients have very kindly got involved with research we've done, donated lots of samples. We've taken to the lab. We've tried to work out what's going on. How do we translate this into new treatments? In fact, I think we're in an era now where these single gene variations are being translated into new treatments. So I mentioned LRRK2. I mentioned LRRK2 inhibitors, kinase inhibitors that turn off the enzyme activity. We don't, as with all research, at the moment we don't know whether this type of treatment is going to be helpful or harmful. We don't know whether it's going to make the condition worse or make it better. So there will be a placebo-controlled trial where people are randomly assigned either to a dummy treatment or to the active treatments. I think this is important. I think everyone goes into clinical research studies, clinical research trials, hoping that will help them. I think that's a very natural. I think I would, and I have actually gone into a trial hoping that it would help me personally. But of course, we don't really know. If we knew it was the right thing to do, and if we knew it was beneficial, we'd be giving it to everybody. So that's what I kind of often say to people, and that's why we have to have placebo-controlled arms in many of the studies that really prove efficacy in new therapies that come through. So GBA, I mentioned what that's particularly important for is the lysosome. So that's like a waste disposal mechanism in the cell. So we know that in Parkinson's, for many patients, there's a build-up of a sticky protein called alpha-synuclein that builds up in the cell. And obviously, if that's building up in the cell, there are kind of only two reasons that can happen. One is the proteins being made at a higher rate, or it's abnormally sticky, it's sticking together, or the cell is not good enough at clearing away the protein. So it's either making too much waste or not getting rid of waste fast enough. The lysosome is the mechanism that gets rid of waste within cells, and actually GBA seems to be very important for this mechanism. It seems that there is a link between GBA and synuclein, and that patients have this variant have a particular problem with lysosomes, waste disposal mechanisms. So there are several different trials actually now in development or completing early stage, including ambroxol, which is a drug which helps GBA activity, with the aim of helping people with this with problems with their lysosomal function. The third example is the Parkinson gene variant. There are probably this is something we're very interested in. This is only really found in patients with very early onset Parkinson's, so usually with people whose Parkinson's started before the age of 35. It is very rare. There are probably no more than 100 individuals in the UK or 100 families in the UK who have variation in the Parkinson gene. But we know that the Parkinson gene particularly affects mitochondrial function, so particularly affects the energy supply in nerve cells, which has led on to a trial which is going on now, looking at the drug that we're involved with, that's been nurtured with Q Parkinson Trust of EPI-589, which is a drug that helps mitochondrial function. In fact, it's on my next slide. This is on my next slide. The $64 million question is actually whether these types of drugs are only going to be effective for people who carry the gene variant, or whether they'll be effective for all Parkinson's patients. Pragmatically, actually, both the trials that I'm aware of, so the GBA studies and the mitochondrial studies, I'm not too sure about some of the LRRK2 studies that are planned, but pragmatically, most of the studies have included a reasonable number of people with the gene variant and also Parkinson's patients without the gene variant. So it could be that these mechanisms actually are kind of important for all Parkinson's patients, and actually you don't have to have this variant to have the problem, but we don't know that. It may be that only people who carry the gene variant are going to respond really well to the treatment. So pragmatically, these studies are mostly now including both some people with the gene variant and some people without the gene variant. Of course, this the whole business of identifying gene variants that greatly increase your risk of Parkinson's, like LRRK2, raises the possibility of pre-symptomatic treatment. So if we knew that you were destined to develop Parkinson's in 20 years' time, it may be that it would be possible for you to enter a trial or a study which would prevent you from developing it in 20 years' time. So pre-symptomatic treatments. And this is something I think, well, as far as I'm aware, there are not studies directly tackling this at the moment. But as things progress, actually very quickly, I think this will be something we're talking about over the next few years, whether we should be mounting pre-symptomatic studies. And actually this is happening in Alzheimer's. So in the rare Alzheimer's families where Alzheimer's is passed from generation to generation, there are trials now going on for people who carry a gene variant, which means they may not develop Alzheimer's for 10, 15 years, to see if the treatment can prevent the disease from developing. So I think I've mentioned this. So yeah, both the EPI-589 and the Ambroxol studies are recruiting both people with the relevant gene variant from which the discovery was made and people without the gene variant. And I think that's going to be important. Okay. So in terms of kind of how we incorporate genetics into our therapeutics and our research, and how we incorporate this into treatments for people in the future, there are a number of sort of ongoing research studies. I'll just mention a few of these quickly. So these are the ones that I'm particularly involved with. There are lots of other studies that are going on as well. So one study that we're very involved with, led by Donald Grosset in Glasgow, is the Proband or Tracking Parkinson's study. So that's recruited 2,200 people with Parkinson's across the UK from 70 study sites, and patients who are involved with that have donated blood samples and have a comprehensive Parkinson's assessment battery that is carried out on about an 18-month basis. So not only are they assessed once for clinical profile problems they've got, but what happens over time is measured as well. I think this is crucially important for our research now, to actually track what happens to people over time. A lot of the genetic studies we've done in the past have relied on just assessing someone at one time point, but actually what happens to people over time, I think, is very important. People have very different trajectories, what happens over time with the disease. We know actually from drug studies, and we're seeing from these other studies, that a lot of patients, well, not a lot, some patients recruited to studies early stage turn out not to have Parkinson's disease. And I think we have to be upfront about that. I think that so I see one of my other interests is other Parkinsonism conditions which are different from Parkinson's disease. And so I see a fair number of people in whom they've originally been thought to have Parkinson's, but then as time goes on, the doctor say, well, actually, we're not sure that you do have Parkinson's. And then they kind of come to my clinic and we do some assessment and try to consider what condition they've got. And sometimes we make a diagnosis which is of a different condition. And patients often say then, oh, this is great, you're the first doctor who's kind of assessed this and know what's wrong with me. And obviously that's very flattering. But what I always say, well, actually, you've had this for a few years now. It gets much easier as time goes on. If I'd seen you at the very beginning, I may well not have known exactly what was wrong with you. So actually, as time goes on, people develop different symptoms and different features. And actually, the diagnosis kind of becomes a lot clearer after 2 or 3 years. So I think following people over time is really important for understanding how the disease changes over time, but also for being as certain about the condition as possible. I think, very understandably, I think uncertainty is very difficult to deal with for patients in terms of what the condition they've got. But I think we have to be honest about, so for example, in the studies for pramipexole and ropinirole, when people were randomized into those studies, actually it turned out that 10% of people in those studies did not have Parkinson's disease. So there's a 10% false diagnosis rate, and this was in research studies doing Parkinson's research. So I think this is a really important issue, and it's one of the things we're trying to tackle and make better through clinical assessment, genetics, biomarkers, to actually try and be as accurate as possible. So another aspect of things is there's a drive for some treatments to try to treat people as early as possible. And the earliest you try to treat people in the disease course, the more uncertainty there is about the diagnosis. So time is very important in clarifying things. And actually one of the most important things when I see people in clinic is trying to assess how well they respond to levodopa. So most people with Parkinson's respond really well to levodopa treatment. And that's really part of the diagnostic assessment of someone with Parkinson's in terms of how well they respond to levodopa. So as a sort of tracking Parkinson's program is not recruiting new patients as a kind of offshoot from that. We've set up a project called Parkinson's Families Project, which are particularly recruiting people whose Parkinson's developed before the age of 45 or who have a family history of other people affected by Parkinson's. So that, we have about 30 study sites in the UK who are recruiting patients to this. Again, the same thing, systematic clinical assessment, blood samples, 100,000 genomes. So, in fact, one of the really interesting things as well is to look back at clinical trial data. So PD Med was a randomized trial that was led by Carl Clark, people randomized between levodopa, selegiline, rasagiline, or ropinirole, pramipexole, dopamine agonists, and they were followed up over time. In fact, there are DNA samples for all the patients involved in that. So we're looking at people's genetic makeup now and integrating that with the clinical trial data. And that's an incredibly valuable, incredibly rich resource for understanding what's happening with Parkinson's. 100,000 Genomes Project. So this is sequencing the full genome of people with a whole range of different conditions. 50,000 people with what's described as rare conditions. And we were able to get early onset and familial Parkinson's recognised as a rare condition. So that's a subset of Parkinson's, so that's included in the 100,000 Genomes Project. There are about 500 people recruited to that. There are about 700 people recruited to the Parkinson's Families Project. Proband is 2,000 people. PD Med Genetics is about 2,000 people. So we're kind of building up very large numbers now of patients who agreed to involve research and have donated DNA samples. There is SysMed PD, which Phil Campbell studied, look at biochemistry, mitochondrial function. And actually one of the things we're doing with Cure Parkinson's Trust is to set up a DNA bank to store DNA people from this point forward who are involved in clinical trials. So anyone who now is in the PD STAT study and then looking back to the exenatide and GNF study, we are collating DNA samples from people who've been involved in these studies with the aim of integrating that with the clinical trial data. And actually one thing I'd say about that is it's much easier to do it when the study's ongoing than later on. And so I think really that's something that would be usefully included in all Parkinson's studies, is ask people if they're happy to have their genetic makeup looked at. And then the other thing that I've not particularly mentioned is that, which is not really research, but NHS testing is available for Parkinson's. So if you have a family history of Parkinson's or you have very early onset Parkinson's, it's possible to have an NHS test for the 7 or 8 known genes that have a high risk of Parkinson's and have a test done and actually learn something about why your Parkinson's developed. One of the other things that is obviously moving quite quickly is direct and consumer testing. So there are a number of companies, Icelandic and American based, where you can send off a DNA sample to them and receive genetic results back through the post and learn a bit about your genetic status. So this is small, but I think this is, in a way, this is kind of the most important bit. So this is communicating results, genetic research, genetic data. Hopefully there will be a handout available later. These are just things. These are things for discussion. These are kind of comments, if you like, things to think about. And I really value your views on some of these things. And I think that how we take this forward has to be a two-way thing between patients, charities, families, researchers. I think these are kind of, in a way, some of these things are societal issues rather than research issues that we kind of need to decide how to take things forward. So these are a few sort of just comments. So one thing that I feel quite strongly about is that everyone has the right to know their genetic data and genetic status. So that is your personal data. So in a way, I think everyone has their right to know their genetic data and genetic status. So actually with the 100,000 Genomes Project, I think that it will be possible for people to just have a copy of all their genetic data. The problem, the caveat to that is that the interpretation of the data may be extremely complex. So even for genes that we know pretty well, the LRRK2 gene, actually whether you actually get the condition can be very complex to interpret from the data. So I would say that ideally one would be in contact with a healthcare professional, a genetic counsellor, a neurologist or geriatrician with interest in genetics, a geneticist who can talk you through the results and try to put them in some context. I think even for a lot of doctors, actually, understanding some of these things is complex. So if you have the condition, whether you get the disease, that's called penetrance. And there's some really strange things happening. So actually one of the genes that causes dystonia, if you carry that, you have a 1 in 3 chance of getting dystonia. But actually, if you get to age 27 and beyond, you will not get the condition. So the risk period only is up to age 27. So there are really strange things going on with some of these genetic variants, which we don't fully understand. And obviously, if you have one of these variants, I think that you should have counselling about that, and you should be told the current best information about what that variant means. So that makes me somewhat cautious about some of this direct-to-consumer testing. We've had relatives come to our clinic who've sent stuff off, got it back in the post, and have come to, well, I don't really understand. I was very alarmed when I got this information. This is suggesting there might be risk to other people in my family. What should I tell them? What should it all mean? And so, obviously, very happy to discuss that with patients and to provide some sort of context to that. But that makes me a bit cautious about direct-to-consumer testing, to have that happening without any sort of counselling or advice. There's also the business about variants of unknown significance. So a lot of rare variants, we don't know if they cause disease. So currently, knowing your genetic status for Parkinson's doesn't change any of your recommended treatment. So currently, if you have LRRK2 or Parkin or GBA, that doesn't change any of your treatment currently. So some people say to me, what's the point of knowing about this? This is not going to change any of the treatment I'm going to have. So what's the point of knowing about it? That obviously is the tests are available, so you can have them. You have to understand what the outcome is going to be before you have the test, what it all means. But some people say, why have the test done? So in the UK, so research labs, so the work we do in our lab, trying to find out new things, we're not licensed to provide results to people. So only accredited NHS labs can provide results to people. So we can't send out results to people. So knowing your status may change you and your family's perception of the disease. Knowing your status may, and I think will, change your eligibility for clinical trials. So I think it is the case that knowing your gene status will change what trials you're eligible for in the future. I mentioned NHS testing. So predictive testing is, what if my dad's or mum's got Parkinson's and they've got a gene variant? What about me? I've got a 1 in 2 chance of getting it. Should I be tested? So that's predictive testing. And we know that's looking into the future. And actually, if someone said to me, do you want to know in 10 years' time whether you're going to have cancer or heart disease? I don't really want to know. I mean, I don't really want to know what the future holds personally for most things. And so what can I do about it? Well, we'll take plenty of exercise, have a healthy diet, don't drink too much, have a abstemious lifestyle, which I kind of knew I had to do that. I kind of knew I had to do that anyway. So actually knowing what happens in the future is not, if there's nothing specific to do about it. We know from Huntington's disease that people in that situation, about 10% of people want to know what the future holds. So about 90% of people in those families don't want to know what the future holds. And the key question, the real question is, what are we going to do about it? Have we got a trial you can go into? Is there a specific prevention we can give you with the genetic data? And that will change things a lot. So we wouldn't offer predictive things for low-risk genes. That's the NHS basis at the moment. These risk variants, which only very slightly, we wouldn't give people information. We don't really know, well, we definitely don't know what advice to give people about that. And there is a big danger of it causing a lot of anxiety about things without really any benefit to patients at all. The other thing which is important to mention is that negative results, so you have a test, it's normal, that doesn't mean you haven't got a Parkinson's gene, because we haven't discovered all the genes yet. In fact, about 90% of our families, where lots of people are affected, we haven't identified the gene. We think there's lots more to discover, which we're in the process of discovering at the moment. So these are some of the issues to kind of think about, which sort of factor into discussion about things. Pragmatically, what we've done is ask people what they want in studies. So this is kind of our current state of the art of consent. And this changes every year. So we modify this a bit almost every year as we understand more about things. So we ask people what they want to try to cover eventualities. So I'll just quickly, I know I'm running a bit out of time, but I'll just quickly go through this. So if possible, I'd like to be informed of research results that might indicate NHS test could be developed or used related to my condition. So obviously it's crazy if we do a test, if we do some research and identify you've got a gene for which is already an NHS test, we have to tell you about that. You don't have to have the NHS test done, but we would like to tell you about that, assuming you want to know about it. If possible, I'd like to be contact if the research results indicate eligible for future research studies. So that obviously is important. If you have a GBA variant, do you want to be in a GBA study? And we'd like to know about that in the future, even though that's not an NHS test for Parkinson's. Would I like to be contacted for generic results indicate that treatment is available? So if, say, ambroxol turns out to be a fantastic treatment for GBA, potentially we're going to be sitting on data like 500 Parkinson's patients with GBA. Obviously, what we'd like to do is to contact them and say, hey, look, we've got a treatment now that may be effective for you. We don't have this, because we've adapted this consent that's going on, we don't have that consent from everybody, but I think almost ethically we'd be bound to contact people if we knew that there was a treatment which might help them based on the research we've done. But we'd like to check people are happy to know about that. And then lastly, what happens if in the course of our Parkinson's research we discover that your family has a breast cancer gene or a colon cancer gene, which means you need to have breast cancer screening or colon cancer screening, which could be potentially life-saving information for your family. So obviously we want to tell you about that if we can help you. And so the last bit is I'd like to perform research results that might indicate NHS or equivalent tests could be used unrelated to my condition that might help me or my family. And again, and actually going back 20 years, nothing was released to people at all. And actually, there ended up being some crazy situations where people were sitting on information which was helpful for families that wasn't being released. So we like to try and ask people about it. Most people say yes to most of these things because it's really it's kind of common sense. But it has, for ethics committees, it has to be nailed down what we can do about disclosing information and what situations we can disclose information. And then lastly, data sharing. This, as many of you will know, sort of Facebook, Cambridge Analytica. I mean, this new regulation's coming in. This is an incredibly hot area. We would like to share data that we generate from research with other researchers, but we have to ask you whether you're happy for that to happen. So I agree that my data can be sort of database, can be shared with other researchers inside and outside the European area. That won't become so relevant soon. And shared, pooled with really thousands and thousands of samples from people to enable large-scale analysis. So the future of this is making data openly available for as many people as possible to go to it and to make discoveries. The key thing is it's your data. It is your personal information, and you need to be happy that that data is going to be uploaded onto a database somewhere so that bona fide researchers can look into that. And some people are not happy with that, and that's obviously absolutely fine. But we need to be explicit with people what's happening with their data in the future, and for people to really understand that and to buy into that to enable the best discoveries in the future. So plan of action, interact with patients and families, make sure our consent and ethics are fit for purpose, continue some of our large-scale studies, getting our data and sample sharing right, get clinical trialists to work out which patients respond best to which treatments. What I would like to see, and I know Rich and Helen, try to ensure that every clinical trial patient who consents has the opportunity to store a sample to look back on genetics and biochemistry in the future for what we call precision medicine, who responded best to the drug. So that's what I've talked about. Sorry I've gone slightly too long. Thanks very much.
Absolutely.
Professor, that was a lot of information. That was a lot of information. I'm a Parkinson's patient, and there were some words I didn't understand, and it was very interesting, the part that I did understand. The one thing I'd like to question is the question of whether to be tested, and whether to ask for the penetration level at different ages depending on which gene you have. And I agree with you in principle that there's no point in knowing what's going to happen in 10 years' time. There's one big question mark, one big qualification to this, and that is that as a result of that attitude, week in, week out, babies are being born with the gene which could have been avoided with approved PGD test tube
Yeah.
baby IVF processes that will stop the inheritance line.
Yeah.
Now that complicates the assessment
Yeah.
enormously.
Yeah.
To the
So
extent.
again, you know, again, that's this kind of societal issue rather than a medical issue. That's kind of, people have different views about this from ethical, religious standpoints. And again, Richard's an expert in this, but you know, can't say that people shouldn't be tested, but I would say that many of us don't want to know what happens in the future. But actually, the jargon when we're talking to people about this is non-directive counselling. So the idea is you talk through the pros and cons of the whole thing, but then just leave it to people to decide what they want to do. So it's non-directive counselling would be the kind of phrase that we'd use.
Clive, we've got time, literally 2 questions. Clive.
Yeah, I'd just like to ask you briefly about mitochondrial DNA, because that's partly where the things may go wrong.
Yeah.
Parkin, you mentioned, affect the mitochondria, but that's a nuclear DNA, isn't it?
Yeah.
Is there no advantage at looking at the mitochondrial DNA and how it might interact with the main nuclear DNA in abnormalities?
Yeah. So it's in the mitochondria, this powerhouse in the cell. About 90% of the proteins are encoded in the nucleus and about 10% in the mitochondria. There has been quite a bit of work looking at mitochondrial DNA. So that's a bit of DNA you only inherit from your mum, and whether differences in Parkinson's patients that haven't come up with anything definite as yet. There's some, the jargon is haplogroups, so there's some, and actually you can trace people's ancestry from this. So there's potentially some haplogroups, so that's something that everybody has, maybe slightly more common in Parkinson's patients. But I think it's fair to say that's not translated to treatment. And actually, mitochondrial function, I mean, it's a good example of something where there are many different routes to a chemical change. And actually, I'm talking myself out of job here. So why look at the genetics? Why not just look at forget the genetics, just look at the chemical change and don't bother studying the DNA? And that has been challenging. But actually, you can see that there's so many different genes that could influence mitochondrial function from the nucleus that actually it may be quicker just to look at the biochemistry rather than the genes. So. But I think it's a really important area, and I think there's a lot more still to learn.
One final question here.
Yeah, I've got one point to make about genetic footprinting. Obviously, this could extend into prospective employers, and insurance companies, and all sorts of places. Couldn't it? Could be like a Big Brother thing if not careful. That's just a point I'm making, really. But regarding your treatments, are you dealing with people with advanced stages at all, or patient groups with early onset? Or
actually, so some of the studies we're doing are particularly recruiting early stage patients. Actually, in the EPI 589 study, the way that was that for people with typical Parkinson's, they really wanted very early stage patients who hadn't really started very much treatment. But actually, if you had a gene variant, because it's quite difficult to find those patients at the moment, it could be any disease stage because they're really interested in the biochemistry and seeing if the drug changed the biochemistry. So actually, there's slight difference, but it varies across studies. So some of the new antibody studies that are starting, again, are going to be targeting earlier stage patients. So that's
a lot of it cost-driven as well, I guess.
Yeah, there are lots of. It's a good question whether this is a good strategy to target people with early stage disease or not. And I think, of course, we don't really know yet because we haven't got a absolutely true bill disease-modifying treatment that stops the disease in its tracks. From the Alzheimer world, the Alzheimer trials, which really target the chemistry of Alzheimer's disease, have been unsuccessful. And it's thought that one of the reasons for that is not targeting people early enough in the disease course. But actually, that's a hypothesis. It's not really proven. And I'm aware there are lots of patients with established Parkinson's who are really keen to be involved in research studies, and certainly I'd encourage people to think about trying to include as many patients as possible in studies. But again, there's an issue of homogeneity of having people at similar disease stage, because in terms of trying to get a readout quickly as to whether something's effective. But in many ways, Roger's a much bigger expert in all this than I am, so I'm sure we'll be happy to tackle some of these issues in his talk. And again, it's in part of apology for running out. I'm going to be around, so I'm happy to sort of have a chat things later on. So,
Hugh, thank you very, very much.